Good deal. Thank you, sir. Hello, this is Dr. Steve Morris. I am continuing with the lecture series for Anatomy and Physiology 1. We'll continue today with the chemical and cellular organization. We had the introductory lecture for chemical and cellular organization done in the previous lecture, and this is part 2 of that same lecture. In the previous lecture series, we described the cell membrane and some of the outer components of the cell membrane, which included carbohydrate structures, protein structures, and lipid structures. We also talked about the phospholipid bilayer, its organization as far as the cell. Well, today what we'll do is discuss the components that are inside the cell membrane and how they affect the cellular anatomy and the physiology involved in the cell as well. First structure we'll talk about is cytoplasm. Cytoplasm is a general term for the material located between the cell membrane and the membrane surrounding the nucleus. A colloid with a consistency that varies between that of a thin maple syrup and an almost set gelatin. Cytoplasm contains many of the proteins that does extracellular fluid. Cytosol, or intracellular fluid, contains dissolved nutrients, ions, soluble and insoluble protein, and waste products. Organelles are structures that are suspended within the cytosol and perform specific functions within the cell. Organelles are the internal structure that perform most of the tasks needed to keep a cell alive and functioning normally. Each organelle has specific functions related to cell structure, growth, maintenance, and metabolism. Cellular organelles can be divided into 2 broad categories. First category is non-membranous organelles that are not completely enclosed in membranes and all of their components are in direct contact with the cytosol. The second type of cellular organelles are membranous organelles. They are isolated in the cytosol by phospholipids membranes, just as cell membranes isolate the cytosol from the extracellular fluid. Some examples of non-membranous organelles include the cytoskeleton, microvilli, centrioles, cilia, ribosomes, and proteasomes. Now, you must be familiar with the types of non-membranous organelles. Examples of membranous organelles include the endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria, and also the nucleus. The cytoskeleton functions as the cell skeleton. It provides internal protein framework and gives the cytoplasm strength and flexibility. The cytoskeleton of all cells includes microfilaments, intermediate filaments, and microtubules. Again, you should be familiar with these structures of the cytoskeleton. This is an illustration that is out of your textbook. It gives you an illustration of the various components of the cytoskeleton. Microfilament, terminal web, again, portions of the cytoskeleton, the intermediate filaments, and then the microtubules. You see it in the illustration, and you see another picture of it in electron microscopy, again, illustrating the cytoskeleton. Microfilaments are the most fragile of the cytoskeleton elements. The actin filaments are basically protein strands, generally less than 6 nanometers in diameter, composed of the protein actin. Actin filaments are common in periphery of the cell. They line the intestinal tract, the terminal web, just inside the cell membrane, and exposed surfaces of the cell. So again, actin filaments, again, protein component, and we'll talk about actin filaments and their role in other cells, specifically the muscle cells, when we go through the muscle system. Major functions of microfilaments: anchors the cytoskeleton to integral proteins of the cell membrane, provide additional mechanical strength to the cell, attach the cell membrane to enclose cytoplasm. Microfilaments also provide interacting with other proteins, determines the consistency of the cytoplasm, and the consistency may vary depending on the region of the cell. Actin can interact with the protein myosin to produce active movement of portions of the cell to change the shape of the entire cell. Again, when we talk about the muscle system, you'll see the importance of actin and myosin in those cells, and we'll describe it in a lot more detail. Intermediate filaments strengthens the cells and help maintain its shape, stabilizes position of the organelle, stabilizes position of the cells with respect to surrounding cells throughout specialized attachment to the cell membrane. Microtubules. All our cells contain microtubules, hollow tubes built from globular protein tubulin. Largest component of the cytoskeleton, the microtubule array in a cell, is centered near the nucleus in a region known as the centrosome. From the centrosome, microtubules extend outward into the periphery of the cell. Number and distribution of microtubules present in the cell can change over time. Functions of the microtubules, and you should know the functions of the microtubules. Primary component of the cytoskeleton, giving the cell strength, rigidity, anchoring positions of major organelles. The disassembly of microtubules provides a mechanism for changing the shape of the cell, perhaps assisting in the cell's movement. During cell division, microtubules form a spindle apparatus which distributes the duplicated chromosomes to opposite ends of dividing cells. We shall consider this process in more detail in later in the lecture series. Microtubules form structural components of organelles such as centrioles and cilia. Thick filaments, they appear only in muscle cells where they interact with actin filaments to produce powerful contractions. Microvilli, many cells have small finger-like or finger-shaped projections of the cell membrane, their exposed surfaces. The microvilli greatly increase the surface area of cells exposed to an extracellular environment. Centrioles. All animal cells capable of undergoing cell divisions contain a pair of centrioles, cylindrical structures composed of short microtubules. The microtubules form 9 groups, 3 in each group. Each of the 9 triplets is connected to the nearest neighbor on either side. During cell division, the centrioles form the spindle apparatus associated with the movement of DNA strands. Centrioles are intimately associated with the cytoskeleton. This is an arrangement of the microtubules. Again, by illustration, you see 3 in groups of 9. This is a view from above that shows you, again, the arrangements, the 3 in groups of 9. Now, mature red blood cells, skeletal muscle cells, cardiac muscle cells, typical neurons have no centrioles. As a result, these cells are incapable of dividing. You should be aware of this fact. Again, the absence of the centrioles prevents these cells from being able to go through cell division. An array. There are no central microtubules. The resulting organization is called an array. An axial structure with radial spokes leading toward the microtubular groups has been observed. The function is not exactly known. Now cilia is relatively long, slender extensions of the cell's membrane, found in cells lining the respiratory tract, cells lining the reproductive tract, and various other locations of the body. Internal arrangement similar to that of centrioles. 9 pairs of microtubules rather than triplets surround a central pair, the array. Cilia beat in a rhythmic fashion, so rhythmically they move fluids and secretions across the cell surface, relatively stiff during the effective power stroke and flexible during the return stroke. Cilia is located in the respiratory tract. Ciliated cells along the trachea beat their cilia in a synchronized way to move sticky mucus and trapped dust particles toward the throat and away from the delicate respiratory surfaces. So along the aspects of the trachea and some aspects of the upper respiratory tree, a mucus layer and the cilia line the surface epithelial cells. When particulate matter from the outside environment, such as dust, such as bacteria, such as viruses, get into the upper respiratory tract, get stuck on this mucus, and then in a wave-like motion, a synchronized wave-like motion, the cilia will expel this mucus from the upper respiratory tract. Now, deep in the respiratory system, the endothelial tissue changes. The tissue lining the delicate surfaces of the lower portion of the respiratory tract does not have the ciliated cells. And because of that, it is more susceptible to viruses and bacteria or other dust particles. So it's important that the ciliated cell structures remain intact in the upper respiratory system. This brings up smoking and the adverse effects of smoking. If cilia are damaged or immobilized by heavy smoking, the tar and nicotine in cigarettes, it becomes a significant problem. Now, there are other metabolic diseases or problems that can also cause a similar occurrence of damage to cilia, but the cleansing action of the cilia is subsequently lost when it becomes damaged, and irritants will no longer be removed from the respiratory system. So in essence, you've lost an essential part of the primary defenses of the respiratory system. As a result, chronic cough, respiratory infections will develop. Now, cilia is also located in the respiratory system. Ciliated cells are also responsible for moving oocytes along the uterine tubes and wafting the sperm or moving the sperm from the testes into the male reproductive tract. So again, we see it in the female reproductive tract where the female eggs, the oocytes, are moved along the uterine tubes by cilia, and again the sperm in the male reproductive tract along the levels of the vas deferens, which is the ductal system in the male reproductive tract. You'll see cilia moving the sperm along those tracts. Now, we will discuss the reproductive system in a lot more detail in this portion of anatomy and physiology. The respiratory system we'll discuss in a lot more detail in the next semester when you'll be studying the respiratory system in more detail. Now, some other intracellular organelles. Talk a little bit about ribosomes. Ribosomes are proteins that are produced within the cells using information provided by the DNA of the nucleus. Ribosomes are organelles responsible for protein synthesis. A number of ribosomes in particular cells varies with the type of cell and the demands of new proteins. This is an illustration from your textbook of a ribosomal subunit. You can see two, a small ribosomal subunit and a large ribosomal subunit. Two types of functional ribosomes exist. Free ribosomes are scattered throughout the cytoplasm. Fixed ribosomes, this is where the proteins that they manufacture will enter into the cytosol, attached to the endoplasmic reticulum. Proteins manufactured by fixed ribosomes enter the endoplasmic reticulum, where they are modified and packaged for secretions. Now, proteasomes are organelles that contain an assortment of protein-digesting enzymes or proteases. These will be very important later on in this lecture, and we'll talk about what the proteases do. But these are basically enzyme aggregates to form a hollow cylinder with an active site facing the inner chamber. Special regulatory proteins block the end of the cylinder and prevent entry of normal intracellular proteins. Cytoplasmic enzymes attach chains of ubiquitin, which is basically a molecular tag, to the proteins destined for recycling. Tagged proteins are quickly transported into the proteasome. Once inside, they are rapidly disassembled into amino acids and small peptides, which can be released in the cytoplasm. Proteasomes are responsible for removing and recycling damaged or denatured proteins and from breaking down abnormal proteins such as those produced within the cells infected by viruses. They also play a key role in the immune system. Now, the endoplasmic reticulum, another organelle, the network of intracellular membranes connected to the nuclear envelope which surrounds the nucleus. The endoplasmic reticulum has four major functions. First is synthesis. Specialized regions of the endoplasmic reticulum synthesize proteins, carbohydrates, and lipids. Storage. The endoplasmic reticulum can store synthesized molecules or material absorbed from the cytosol without affecting the cellular operation. Transport. Materials can travel from place to place in the endoplasmic reticulum. And then finally, detoxification, an important function of the endoplasmic reticulum. Drugs or toxins can be absorbed by the endoplasmic reticulum, neutralized by the enzymes within it. Now, you're looking at an illustration from your textbook which demonstrates the endoplasmic reticulum. And there are two types of endoplasmic reticulum. There is the rough endoplasmic reticulum, where you can see the ribosomes have attached to the surface of the endoplasmic reticulum, giving it a rough appearance. And then there is the smooth endoplasmic reticulum that does not have ribosomes attached. Now, the rough endoplasmic reticulum, because it has the ribosomes attached, is responsible for protein synthesis. That's important, and you should know that. The smooth endoplasmic reticulum, because there are no ribosomes associated with it, responsible for carbohydrate production and also responsible for lipid synthesis as well. You can see in the electron microscopy slide, which is next to the illustration, again the pattern of the rough endoplasmic reticulum, where you can see the small ribosomes are attached to the surface of the endoplasmic reticulum, and then the smooth endoplasmic reticulum, which is adjacent to it, have no ribosomes attached. Thus, rough endoplasmic reticulum, smooth endoplasmic reticulum. And again, to repeat, smooth endoplasmic reticulum, no ribosomes. Smooth endoplasmic reticulum has a variety of functions. All of those functions are associated with the synthesis of lipids and carbohydrates. Function of the smooth endoplasmic reticulum: synthesizing the phospholipids and cholesterol needed for the maintenance and the growth of cell membranes, endoplasmic reticulum, nuclear membranes, Golgi apparatus, and all cells. Synthesizing steroid hormones such as androgens and estrogens, which are the dominant sex hormones in males and in females, respectively, in the reproductive organs. Synthesizing and storing glycerides, especially triglycerides in liver cells and fat cells, and then synthesizing and storing glycogen in skeletal muscles and liver cells. The smooth endoplasmic reticulum, synthesizing the phospholipids and the cholesterol needed for the maintenance and growth of cell membranes, endoplasmic reticulum, nuclear membrane, Golgi apparatus, in all cells. This slide is a little redundant. Again, synthesizing the hormones, synthesizing and storing glycerides, and then synthesizing and storing glycogen. Glycogen, especially in skeletal muscle and liver cells, we alluded to the molecular structure of glycogen early in the lecture series. Understand that glycogen is an important source of energy in the system. Glycogen is broken down in the system into glucose, a 6-carbon sugar that's very important for cellular metabolism. So you can see the importance of the relationship with the smooth endoplasmic reticulum. The rough endoplasmic reticulum functions as a combination of workshop and shipping depot. Newly synthesized proteins undergo chemical modification. Proteins are packaged for export in their next destination, the Golgi apparatus. Fixed ribosomes. Ribosomes on the outer surface of the rough endoplasmic reticulum are fixed ribosomes. Their presence gives rough endoplasmic reticulum a beaded, grainy, rough appearance. Both free and fixed ribosomes synthesize proteins by using instructions provided by messenger RNA. Most of the proteins and glycoproteins produced by the rough endoplasmic reticulum are packaged into small membranous sacs that pinch off the tips of the cisternae. Now, these transport vesicles subsequently deliver their contents to the Golgi apparatus. The Golgi apparatus transports vesicles carry newly synthesized proteins or glycoproteins that is destined for transport from the cell. These travel from the endoplasmic reticulum to an organelle that looks a bit like a stack of dinner plates. I don't know about that analogy. I'll let you make that determination where they actually look like dinner plates or not. but that is what's referred to as the Golgi apparatus. As you can see in this illustration and the electron microscopy that's taken from your textbook, you can see this pattern. It looks better in the electron microscope where it gives an appearance of a stack of dinner plates. Now those are kind of unsteady dinner plates if you ask me, but I can see the similarity when that analogy is given. In the illustration, I think it's a little more representative of the surface of this structure, especially as you can see the secretory vesicles coming off the maturing face of this structure. Now, in the subsequent slide, we'll talk about a little bit about the packaging of these particular enzymes and vesicles that form out of the Golgi apparatus. Now, the Golgi apparatus basically has three major functions. You should be familiar with the three major functions. It modifies and packages secretions such as hormones or enzymes, releases it through exocytosis. It renews or modifies the cell membrane. It packages specialized enzymes within the vesicles used in the cytosol. Now, if you look at the illustration in your textbook on the Golgi apparatus, there are two phases you should be familiar with, a forming phase and a maturing phase. The forming phase, as you can see, receives vesicles from the endoplasmic reticulum. These vesicles, which have a phospholipid membrane, because they are membranous organelles, will fuse with the Golgi apparatus, which also is a membranous organelle, and the two membranes will fuse. Once these contents of the vesicles are released into the Golgi apparatus, a processing will take place in this structure. As the processing is completed, in the maturing phase, vesicles are again released. These vesicles are released, which will carry out whatever their specific particular activity need to be, especially in the form of lysosomes, and I'll go into more detail about that. These secretory vesicles will either be released inside the cytosol or will move to the cell membrane. And again, if you remember, the cell membrane is a phospholipid bilayer, and this secretory vesicle, because it is a phospholipid membrane, will fuse to the cell membrane and through a process referred to as exocytosis will expel its contents outside of the cell. So, transport vesicles, and that's what we just described. Transport vesicles can then deliver products to the Golgi apparatus. The vesicles generally arrive from the cisternae, known as the forming phase or the cis phase. The transport vesicles will then fuse with the Golgi membrane, emptying their contents into the cisterna. And that's what we're looking at on this side. Again, vesicles released from the endoplasmic reticulum will come into the forming phase. As they fuse, they release their contents into the cisterna. Now, inside the Golgi apparatus, enzymes will modify the arriving proteins and glycoproteins. The example given in your text, enzymes may change carbohydrate structures to glycoproteins, or they may attach phosphate groups such as sugar, fatty acids to proteins. At the maturing phase, three types of vesicles are formed and carry material away from the Golgi apparatus. Secretory vesicles. Secretory vesicles are vesicles containing secretions that will be discharged from the cell. These vesicles fuse with the cell membrane and empty their contents into the extracellular environment. And again, I showed you that on the previous illustration. There are membrane renewal vesicles. These vesicles produced at the Golgi apparatus fuse with the surface of the cell. They are adding new lipids, new proteins to the cell membrane. Remember, the content of the cell membrane, phospholipid bilayer, but also has integral and peripheral proteins, also has carbohydrate components such as the glycocalyx. It also has lipid components such as cholesterol. Again, that was clearly demonstrated in previous lectures about the contents of the cell membrane. And so these membrane renewal vesicles will allow the the cell membrane to change its properties again over time. And then finally, the lysosomes. These vesicles contain and remain in the cytoplasm and contain digestive enzymes. Now, let's talk a little bit about the lysosomes. Cells often need to break down and recycle large organic molecules and even complex structures like organelles. The breakdown process requires the use of powerful enzymes and is often generate toxic chemicals that could damage or kill the cell. So because they are packaged within these vesicles, they don't damage the cell. They certainly don't enter into the cytosol until a processing has taken place where they may be rendered harmless. There are three basic conditions that will activate lysosomes. When a primary lysosome fuses with membrane of another organelle, such as mitochondrion. When the primary lysosome fuses with an endosome containing fluid or solid material from outside the cell, thus the name autolysis, and I'll show you an illustration of that. And then the other condition that can activate a lysosome is when the lysosomal membrane breaks down following injury to or death of the cell. Now, this is an illustration again from your textbook that shows lysosomal activity. We've already talked about the process of exocytosis. We haven't talked about endocytosis, but we'll do that now. So, a lysosome, a primary lysosome, is produced at the maturing phase of the Golgi apparatus. And then one of three types of lysosomes can develop. The first of which is when this lysosome will come in contact with a damaged organelle. The example used from your book would say a mitochondria. This mitochondria has to be taken out of the cytosol. It's now considered, say, intracellular debris. And so the membrane of the organelle, if you remember, is phospholipid. So it will fuse with the membrane of the lysosome, which is also phospholipid, and will engulf the contents or this damaged organelle. Then through enzymatic processing, it will be broken down into its subunits, and then those substances can be reused in the system. And so a degree of reabsorption will take place. And then following this processing, the remainder, or whatever the residue from that processing is, will be ejected from the cell by exocytosis. Now again, remember, the membrane, this is a membranous organelle, the membrane is phospholipid, will fuse with the phospholipid bilayer of the cell membrane, and in the process of exocytosis will be ejected from the cell. Another type of lysosome, again, primary lysosome is produced at the maturing phase of the Golgi apparatus. And then when the lysosome is produced, a secondary lysosome is made available to pick up substances that have been brought into the cell. Now, this is a process referred to as endocytosis. Let me give you an example. A bacteria, for example, lies on the surface of a cell. Now, this bacteria is an invader into our system, but it hasn't made it into the cell. But for the cell to destroy the bacteria, one of the processes that would have to take place would be an engulfing of that foreign body, that bacteria. The phospholipid bilayer would basically surround that bacterial agent, that extracellular particle or solid, would bring it into the cell, and because the secondary lysosome has powerful digestive enzymes, it will destroy or break down that substance. It will then go through the process of exocytosis, as I described earlier. Now, that's bacteria. But there are substances that are outside the cell that are actually needed inside the cell. You know, we've talked a little bit about electrolytes already, electrolytes, sodium, potassium, that are required inside the cell. But other components, such as glucose, also may be needed inside the cell as well. Specific receptors, by the process of endocytosis, will bring those substances inside the cell as well. again through a digestive process, digestive enzymes inside the lysosomes, will break down these contents and will allow reabsorption to take place inside the cytosol, and whatever residue is left will still be ejected from the cell in a process of exocytosis. And then the third type of primary lysosome goes through a process of autolysis. Basically, substances are released into the cytosol. Again, these digestive enzymes are released. Some of which would be needed in the cytosol for different cellular activities. So, as a review, enzyme activation. Now, we talked about enzymes earlier in the lecture series. Enzyme activation occurs when lysosome fuses with the membrane of transport vesicles or damaged organelles targeted for destruction, such as mitochondria or fragments of the endoplasmic reticulum. This fusion creates a secondary lysosome which contains active enzymes. These enzymes then break down the lysosomal contents. Nutrients re-enter the cytosol, and the remaining material is eliminated from the cell by exocytosis. Lysosomes also function in defense against disease. Cells may remove bacteria as well as liquids or organic debris from extracellular fluids by enclosing them in small portions of the cell membrane, which is then pinched off to form a vesicle endocytosis. Lysosomes then fuse with vesicles. Lysosomal enzymes break down the contents of the lysosomes and release usable substances such as sugars or amino acids. In this way, the cell both protects itself against pathogenic organisms and obtains valuable nutrients. Now, there are some diseases that specifically affect the lysosomes. These are referred to as lysosomal storage diseases. I won't go into a lot of detail about them now, but in another lecture series, we will talk about lysosomal storage diseases. Problems with lysosomal enzyme production cause more than 30 serious diseases affecting children. Lack of specific lysosomal enzymes result in a buildup of waste products and debris normally removed and recycled by lysosomes. Affected individuals may die when vital cells, such as those of the heart, can no longer function. Peroxisomes. Smaller than lysosomes and carry a different group of enzymes. Produced by growth and subdivision existing peroxisomes, their enzymes are produced as free ribosomes and transported from the cytosol into the peroxisomes by carrier proteins. Peroxisomes absorb and break down fatty acids and other organic compounds. Peroxisomes generate hydrogen peroxide, a potentially dangerous free radical. Other enzymes within the peroxisomes then break down hydrogen peroxide to oxygen and water. Peroxisomes thus protect the cell from potentially damaging effects of free radicals produced during catabolism. Peroxisomes numbers are typically highest in metabolically active cells, such as liver cells. That's important. Again, we won't go into a lot of detail about the liver and the hepatic system right now, but later in the lecture, in A&P Part 2 rather, we'll go through the digestive system and the liver, one of the major organs in the digestive system that's involved in detoxification, and you'll see why that's important, that the number of peroxisomes is highest in an organ system such as that. Another concept that you should just appreciate is membrane flow. Please read that in your text. Basically, dynamic nature of cells provides a mechanism in which cells can change the characteristics of their cell membrane, lipids, receptors, channels, anchors, enzymes as they grow, mature, and respond to specific environmental stimuli. Now, another very important organelle in the system is called the mitochondria. And we'll go a little bit into detail about the mitochondria at this point. Now, the primary responsibility of the mitochondria is energy production. Mitochondria absorb short-chained carbon chains, such as pyruvic acid and oxygen, and generates carbon dioxide and ATP. Now, if you remember earlier in the lecture series, we talked about glucose and the ability of glucose to provide energy, but for glucose to provide that energy, it has to be broken down glucose is a 6-carbon sugar, has to be broken down into a 3-carbon sugar, pyruvic acid, in a process referred to as glycolysis. When glycolysis occurs, it breaks glucose into a 3-carbon sugar. The 3-carbon sugar, pyruvic acid, is then moved into the mitochondria where it goes through a process referred to as the Krebs cycle, the TCA cycle, tricarboxylic acid cycle, or the citric acid cycle. It generates, in the presence of oxygen, carbon dioxide and ATP, adenosine triphosphate, which is a very powerful energy source in the body. This illustration taken from your book shows you those basic steps. Again, beginning with glucose. And remember, we talked about glycogen earlier, which is stored in muscle, stored in the liver. A source of energy. Glycogen is broken down into glucose. Glucose moves inside the cytoplasm through a process referred to as glycolysis. Glucose is broken down into a 3-carbon pyruvic acid. Pyruvic acid then moves into the mitochondria, goes through the TCA cycle. The TCA cycle is basically a series of enzymatic steps which will generate an energy packet, ADP, adenosine diphosphate. Phosphorylation will take place. The production, again in the presence of oxygen, will produce ATP. ATP being a powerful energy unit. Now, this is a very important cycle. It's also important for you to recognize that glycolysis takes place in the cytoplasm. TCA cycle takes place inside the mitochondria. Oxygen is needed. This is an aerobic process, so oxygen is required. Phosphorylation will occur to produce ATP. The cristae basically structures inside the mitochondria, increase the surface area of the mitochondria. Again, just going back to this basic structure, you can see the cristae here inside the mitochondria. And because of the redundant folds that are seen inside the mitochondria, it basically increases the surface area of the mitochondria inside. So we see surface area exposed to fluid contents. The matrix have an increased surface layer based upon the role of the cristae in the mitochondria. Now, the membranes of the mitochondria, the outer membrane surrounds the organelles. The inner membrane contains numerous folds referred to as cristae, as we've already alluded to. There are metabolic enzymes of the mitochondria. Metabolic enzymes in the matrix catalyze reactions that provide energy and cellular function. Most of the chemical reactions release energy occur in the mitochondria. Most of the cellular activity that requires energy occurs in the surrounding cytoplasm. Cell energy. Cells must therefore store energy in a form that can be removed from place to place. Cells can break the high energy bond under controlled conditions, reconverting adenosine triphosphate, ATP, to ADP, adenosine diphosphate, and phosphate is thereby releasing energy to the cells for use. An important aspect of ATP. Now, ATP production occurs inside the mitochondria. We've already looked at that. First step takes place in the cytosol. We also looked at that as well. Again, that first step is breaking down glucose into pyruvic acid. That process, again, glycolysis. Each glucose molecule, which is a 6-carbon molecule, broken down to 2 molecules of pyruvic acid. Each of those 2 molecules are 3-carbon components. The pyruvic acid molecules are then absorbed by the mitochondria. In the mitochondrial matrix, a molecule is removed from each absorbed pyruvic acid molecule. The remainder enters the tricarboxylic acid cycle, or the TCA cycle, also known as the Krebs cycle, also known as the citric acid cycle. You should be familiar with those terms. The TCA cycle or the Krebs cycle is an enzymatic pathway that systemically breaks down the absorbed pyruvic acid and the presence of oxygen. Now, I'm not going to ask you to know each of the steps of the TCA cycle, so I don't necessarily want you to spend a tremendous amount of time trying to memorize each of the steps of the TCA cycle, and it is described in your text. The important fact for you to understand is this is an enzymatic process. Pyruvic acid goes through several steps of metabolism through this enzymatic process, but the end result is the generation of that energy source, ATP. And again, the location where the TCA or the Krebs cycle takes place is inside the mitochondria. Aerobic metabolism, I've also already mentioned that this is done in the presence of oxygen. Oxygen is required. So mitochondrial activity requires oxygen. The method of ATP production is known as aerobic metabolism or cellular respiration. Aerobic metabolism in the mitochondria produces 95% of the ATP needed to keep the cell alive. Now that's a key point. 95% of ATP needed to keep the cell alive is produced in aerobic metabolism in the mitochondria. Now the other 5%, which we won't go into detail about right now, but the other 5% is produced by anaerobic metabolism, which is in the absence of oxygen. Now that's important as well, and actually the production of pyruvic acid is done in an anaerobic type of metabolism, so obviously we do need anaerobic metabolism as well. However, for the cell to live, 95% of ATP production, for the cell to live, aerobic metabolism in the mitochondria is required. The other organelle that we'll talk about is the nucleus. The nucleus actually is a centrally located structure in most cells. I can't say that in all cells, but in most cells it's a centrally located structure that typically takes a dark stain, and so when you see it under a microscope, you see an intensely stained dark central area referred to as the nucleus. You can see that both on the illustration to your right, and then you can also see that under electron microscopy. The nucleus is usually the largest, most conspicuous structure in a cell, serves as the control center of cellular operations, stores all the information needed to direct synthesis of more than 100,000 different proteins in the human body. It determines the structure of the cell, what the function it will be performing by controlling which proteins are synthesized under what circumstances and in what amount. Now, the nucleus is surrounded by a nuclear envelope. So surrounding the nucleus and separating it from the cytosol is a nuclear envelope, a double membrane that has two layers separated by a narrow perinuclear space. Remember, the environment inside the nucleus is exclusively different from the environment that's outside the nucleus. That environment outside the nucleus is the cytosol or the cytoplasm. And so there has to be some communication between the nucleus and the cytosol because the nucleus obviously needs energy sources, protein elements as well. And so that communication takes place through structures referred to as nuclear pores. This is the chemical communication between the nucleus and the cytosol. It basically covers about 10% of the surface of the nucleus. It's large enough to permit the movement of ions and small molecules, but it's too small for free passage of proteins or DNA. Now remember, information for protein production still has to get in and out of the nucleus, and so little messenger RNAs are responsible for that because the DNA is actually too large to pass out of these little nuclear pores. Also, again, protein structures are typically too large as well. So, again, other mechanisms have to be set inside the nucleus for information and the obtainment of nutrient sources and the removal of waste has to be set up. And so the nuclear pores, though, is that chemical communication. Regulatory proteins that govern the transport of specific proteins and RNA into or out of the nucleus are set up. Fluid contents of the nucleus are basically called nucleoplasm. Most nuclei contain several dark-stained areas referred to as nucleoli. Nucleoli are transient nuclear organelles that synthesize ribosomal RNA. Nucleoli composed of RNA, enzymes, and proteins are called histones. And then DNA in the nucleus that store the instructions of protein synthesis. Then this DNA is organized into a structure called chromosomes. Chromosomes, nuclei of somatic cells, contain 23 pairs of chromosomes. One member of each pair is derived from the mother and one from the father. Each chromosome contains DNA strands bound to histones. This illustration is out of your book, and it just basically shows you the coiling that can take place using these histone structures. Now, there is a powerful attraction between the histone and the DNA strands. Because of that powerful attraction, the DNA strands literally will wrap around the histones. And if you follow this process, if you look at the DNA structure, which is a double helix, and we've described and we've alluded to this earlier in the lecture series when we were talking about complementary base pairing, but you can see how this double helix will begin to coil around the histone and eventually will wrap up and develop what's referred to as a supercoiling. And because of this supercoiling structure of DNA, a tremendous amount of information can be packed into the nucleus because of this supercoiling. So, histones, the DNA strands coil around the histone. This allows a great deal of DNA to be packaged into small spaces. Interactions between DNA and histones help determine the information available to the cell at any moment. DNA strands wind around the histones, forming complexes known as nucleosomes. Now, the genetic code. Each protein molecule consists of a unique sequence of amino acids. This is basically a recipe for proteins. Therefore, it must be specific, or there must be a specific, or you must specify the order of the amino acids in the polypeptide chain. The information stored in the chemical structure of the DNA strand is referred to as the nucleus. The gene is a functional unit of heredity and contains all the DNA triplets needed to produce protein. The triplet code, a single DNA molecule consists of a pair of DNA strands held together by hydrogen bonding between complementary nitrogenous bases. A sequence of 3 nitrogenous bases specifies the identity of a single amino acid. Information is stored in a sequence of nitrogenous bases along the length of the DNA strand. Those nitrogenous bases are adenine, thymine, cytosine, and guanine. And I know I've alluded to these earlier in the lecture series when we were talking about the bases several lectures ago. A gene is a functional unit of heredity, contains all the DNA triplets needed to produce specific proteins. The number of triplets varies from gene to gene depending on the size of the polypeptide represented. Instructions for transfer RNA. Not all the DNA molecules are devoted to carrying instructions for proteins. Some segments contain instructions for the synthesis of transfer RNA or ribosomal RNA, and others have no function. Messenger RNA involved in transcription. A small portion of a single DNA molecule containing a single gene is available for transcription. This illustration is taken out of your book. Again, this is the DNA. Already familiar with this structure. Now, again, this is now given on one plane, but remember this is a coiled double helix structure, as you can see here. There are enzymes that will help uncoil this double helix structure, and we'll talk about some of those enzymes in just a bit. But for replication to take place, these complementary base pairs, the double helix, have to be separated. A template has to be laid down, again, of complementary base pairs. It has to match up. But new segments are laid down on each side. This picture basically just illustrates this reproductive process, this division process on just one side or one chain of this double helix. But remember, the similar process is going on the other side. So as the complementary base pairs are stripped apart, new material is laid down on one complementary base pair, and the same process is going on the opposite side. Typically in chains of 3, referred to as codons, and we'll go into those in just a little bit. But this is just a basic illustration showing you this process. So, in the steps, messenger RNA and transcription. So, step 1 of this transcription process, 2 DNA strands will separate. The RNA polymerase, which is an enzyme, binds to a promoter site. A promoter site is basically a sequence of these bases that tells the RNA polymerase where to start at, where to split the chain at its beginning. So the RNA polymerase moves from one nucleotide to another along the length of the template strand. At each site, complementary RNA nucleotides form hydrogen bonds with the DNA nucleotides of the template strand. The RNA polymerase then strings the arriving nucleotides together in a strand of messenger RNA. Step 3, on reaching the top of the signal at the end, the stop signal basically will tell this process to end, the RNA polymerase and the messenger RNA strand will detach and two DNA strands will reassociate. Now, there's a coding strand. Two DNA strands in a gene are complementary. And you remember that from, again, how the base pairs will line up. Okay, so the two strands must be complementary. One of the two strands contains a triplet that will specify the sequence of amino acids in the polypeptide. The resulting messenger RNA will have a nucleotide sequence identical to that of the coding strand, but with uracil rather than thymine. The template strand contains complementary triplets that will be used as a template for messenger RNA production. A codon, a 3-base messenger RNA sequence, is called a codon. It contains nitrogenous bases that are complementary to those of the triplets in the template strand. For example, if a DNA triplet has thymine, cytosine, and guanine, the corresponding messenger RNA codon will be adenine, guanine, and cytosine. Now, this method of copying ensures that the messenger RNA exactly matches the coding strand of the gene. This is so important. Now, the RNA polymerase. RNA polymerase interacts with only a small portion of the template strand at any one time as it travels along the DNA strand. The complementary strand separates from the enzyme as it moves one nucleotide at a time and then reassociates behind it. The enzyme collects additional nucleotides and attaches them to the growing chain. Now, we've already talked about the substitution of uracil for thymine. Nucleotides involved in those characteristics of RNA, not in DNA. RNA polymerase attaches adenine, guanine, cytosine, or uracil, but never thymine. So, wherever you see adenine occurs in the DNA strand, the polymerase will attach uracil rather than thymine to the growing messenger RNA strand. And this illustration, again, out of your book, again, simplifying the process of transcription, especially in the nucleus, and how it produces a strand of messenger RNA. The sequence of messenger RNA codon determines the sequence of the transfer RNA and thus the sequence of amino acids. Ultimately, amino acids have been strung together to form polypeptides. The transfer RNAs have been released and the messenger RNA strands remain intact. Now, transfer RNA, the amino acids are provided by a transfer RNA, relatively small mobile types of RNA. Each transfer RNA molecule binds to delivers amino acids in a specific type. More than 20 kinds of transfer RNA exist, at least one for each of the amino acids used in protein synthesis. Transfer RNA molecule has a tail that binds an amino acid. Roughly midway along the length of the nucleotide chain of transfer RNA forms a tight loop. The loop that can interact with the messenger RNA strand contains 3 nitrogenous bases that forms an anticodon. During translation, the anticodon will bond complementarily with an appropriate messenger RNA codon. The base sequence of the anticodon indicates the type of amino acid carried by the transfer RNA. These are some examples of triplet codes. Now, I am not going to ask you to give me these triplet codes for coding strand, the triplet code for template strands, or the triplet code for messenger RNA codon or anticodon for transfer RNA. What I do want you to appreciate is just typically how each of these coding strands have specific base pairs. They must be complementary, and there are specific amino acids that are associated with them. Again, I'm not going to ask you the specific coding patterns. I'm not going to ask you the specific amino acids that relate to them. I just want you to have an overall appreciation for it. Now, the process of translation, the messenger RNA strand binds to small ribosomal subunits. It's used to join the start codon by the first transfer RNA, which carries the amino acid methionine. Binding occurs between the complementary base pairs of the codon and the anticodon. And then in the second step, a small, large ribosomal subunit interlock around the messenger RNA strand. Now, because the base pairing sequence is so important, if anything alters that sequence, it can certainly affect the cell's DNA. So this brings up the point of mutation. Permanent changes in a cell's DNA that affect the nucleotide sequence of one or more genes is referred to as a mutation. Point mutations, a change in a single nucleotide that affects one codon. Production of a codon that specifies a different amino acid usually changes the structure of a complementary protein. A single change in the amino acid sequence of a structural protein or enzyme can prove fatal. Certain cancers and two potentially lethal blood disorders, thalassemia and sickle cell anemia, result in variations of single nucleotides. Cell division. An amazing transformation is a form of cellular reproduction. The division of a single cell produces a pair of daughter cells, each half the size of the original. Before dividing, each of the daughter cells will grow to the size of the original cell. Now, the process referred to as mitosis. This is a duplication of the cell's genetic material, again called DNA replication. Okay, nuclear division is called mitosis. Mitosis occurs during the division of somatic cells. And it's important to get to understand the difference. Again, the cell's genetic material, which is in the nucleus, referred to as DNA replication. Again, nuclear division is called mitosis. Mitosis occurs during the division of somatic cells. Meiosis, the production of sex cells, involves a different process. Meiosis we will go into more when we describe the reproductive system. Now, this illustration is taken from your book. It goes through the phases of mitosis. Again, this is nuclear division. We'll go through each of these phases a little more in detail. Somatic cells spend the majority of their functional lives in a state known as interphase. That's important to understand. Again, the majority of somatic cells spend the majority of their functional lives in a state known as interphase. During interphase, the cell performs all of its normal functions and, if necessary, prepares for cell division. Now remember, in interphase, it only prepares for cell division. It's not actively going through cell division at this point. Okay? Phase that we'll talk about first involves this same process as illustrated here. Okay, so the G1 phase, a cell is ready to divide. It first enters into this phase, makes enough mitochondria, cytoskeleton elements, endoplasmic reticulum, ribosomes, Golgi membranes, and cytosol for two functional cells. Okay? Centriole replication can begin. Now again, this is the G1 phase. Normal cell function plus growth, duplication of organelles, protein synthesis, getting ready for the division to take place. That's the G1 phase. Now, in the S phase, when activities have been completed, the cell enters this S phase. Over about a 6 to 8 hour period, the cell duplicates its chromosomes, involved in DNA replication and synthesis of histones and other proteins in the nucleus. Okay, going back to our illustration again, this is the S phase. Okay, the next phase that we'll see is the G2 phase. Once DNA replication is ended, there's a brief 2 to 5 hour period of time devoted to the last minute of protein synthesis and to the completion of centriole replication. Okay, that's the G2 phase. And then in the M phase, that is where mitosis will begin. In the M phase, again shown on the illustration. So those are the phases. G1 phase, normal cell function plus cell growth, duplication of organelle, protein synthesis. The S phase, DNA replication, synthesis of histones. G2 phase, additional protein synthesis. And then, of course, the M phase. And we'll go into a little more detail about the M phase and the subphases that are involved in this process.